So brain damage is devastating, and the reason is that in contrast to many other tissues in the organism, neurons, once they are damaged and lost, they can't regenerate. And this was a notion that is well known to us since long time. A Spanish neuroscientist, more or less 100 years ago, Santiago Ramón y Cajal, noted that everything may die, nothing may be regenerated. And however, he made the very important statement that it's for the future, for the science of the future, to change, if possible, this harsh decree. So the question is really, is it possible to change this harsh decree? And this is something where we are working hard to make our contribution. So the light at the end, the light at the end of the tunnel is somehow actually provided to us by developmental biology, fundamental developmental biology, which has led to the recognition that virtually all nerve cells, all neurons in the brain are generated directly or indirectly from cells of glial origin, of glial identity. And these cells are referred to as radial glia, and we can call them also maybe stem cell glia for simplicity. These stem cells give rise to neurons during development, but of course they dry out as we become adult, and essentially there are very few regions in the adult brain where regular stem cells would be resident. Now, however, there are, even after pathology. So even after pathology, of course, we don't have any cells that would give rise to neurons, but these cells are surrounded by parenchymal glial cells that have many features in common with stem cells. And the interesting idea that arises from this observation is that could these glial cells, like astrocytes, be a source for new neurons? And again, developmental biology gives us some clue how one could eventually think of this problem by demonstrating that, contrary to what we have believed for a long time, cell identity is not irreversibly fixed, but eventually can be altered. And this was, of course, work that has been pioneered by John Gurdon decades ago, where he showed that a somatic cell can eventually acquire any new identity if one knows a strategy to allow this cell to get there. So these are very fundamental insights from developmental biology, which we now want to use in attempting to convert local glial cells within the brain to neurons. And so the idea is, can we reprogram glial cells into new neurons to regenerate missing cells? Now, the way that one can do this is of course making use of first model systems, and here's our favorite model system, the royal mouse. So the idea here is that we introduce genetic information that encodes for master regulators of cell fate into the glial cells, the astrocytes, and by introducing this information, we alter the gene expression programs active in these cells such that they would actually convert into neurons. And these pictures are actually reflecting what we observe in the mouse in vivo. So this is actually feasible, and the important questions that we now want to address is how similar do these induced neurons become to the endogenous neurons? Because of course, that would be very critical if these cells should be able to fulfill functions within the brain. And of course, this is connected to the question, can these induced neurons integrate into the circuitry, and would eventually these integrated new neurons restore lost function? So these are the problems that we are currently actively researching, and we also try now actually to humanize the mouse model in a way that we can replace the glial cells from the mouse by human cells, so that we can actually test whether a human astroglial cell can be reprogrammed into a neuron in an in vivo context. I just want to show you maybe two more pictures of cells that we have obtained by this approach. So this shows you that these actually look like good neurons. And eventually, if the image would be, the light would be low, you would even see that these cells can make axons and make connections. And I thank you for your attention.